Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello and welcome to the much anticipated and kind of late June 2020 update on progress in Alzheimer's disease blood biomarkers. My name is Elyn Rowe and I'll be your host for this episode. If you're listening to these as they are released, you'll notice that it's now October and I'm covering papers from June. So sorry about the delay. Like many of you listening, I think, anyway. I'm a graduate student and I just had my hands pretty tied with scholarship applications and trying to scrape together some preliminary data for grants in the time of COVID, which has all been a bit chaotic. But enough about me, so let's talk blood biomarkers. Blood biomarkers are, and have been for a few years now, a really hot topic, and in particular, this field of research is critical in facilitating the diagnosis of Alzheimer's disease in a cheap and non-invasive way. So the topic is also near and dear to me, as I'm in a lab for my PhD that focuses a lot on blood biomarkers, and I've actually integrated some blood biomarker work into my own PhD project. So if you're interested, I actually just had an interview with another podcast called Brain Buzz in a crossover episode that we did with AMiNDR, where I talk a little bit about my own PhD research and about the overall importance of blood biomarkers in distinguishing between different types of dementia. So keep an eye out for the release of that episode for a lighter, more conversational take on blood biomarkers and the field of Alzheimer's disease research in general. But today you're here for a comprehensive update on blood biomarkers for AD. So buckle in. From the month of June, there were 28 papers published on the topic, and they span a range of targets, some of which I thought were pretty novel and interesting, and some that are classic targets that you may be familiar with if you've done some reading about blood biomarkers in AD already. So in this first segment, I have 4 papers that focused on RNA as a blood biomarker. The first one was titled Immune and Inflammatory Pathways Implicated by Whole Blood Transcriptomic Analysis in a Diverse Ancestry Alzheimer's Disease Cohort. This is by first author Griswold and last author Beecham, published in the Journal of Alzheimer's Disease. So this study aimed to identify transcriptional changes in the blood that distinguished AD cases from controls, with an emphasis on the ethnic differences in their cohort. So their cohort consisted of nearly 500 participants, with a balance between AD cases and controls, and also a balance between African American ancestry and non-Hispanic whites. Each participant had blood drawn and RNA sequencing done to yield their protein-coding transcriptome, indicating which genes were present as mRNA in the blood. They analyzed their transcriptome data in stratified groups based on ethnicity, so they analyzed all of those with African American ancestry as one group and all of those as non-Hispanic whites in another group. And they also analyzed them all as one group in their joint analysis, where they looked for interactions with ancestry as well. In the stratified analysis, they found over 400 differentially expressed genes between cases and controls in each ethnic group on their own, but only 16 of these genes from each group overlapped between the African American group and the non-Hispanic white group. When they analyzed it in their joint analysis, all as one group, over 1,000 genes were identified as differentially expressed. In their interaction analysis, they found 27 genes that were differentially expressed in the AD cases versus controls from the African American group, compared to the AD cases and controls in the non-Hispanic white group. An example of one of these genes was the MACROD2 gene, which codes for protein deacetylase. This MACROD2 gene expression was found to be strongly decreased in African American AD cases versus controls, but strongly increased in non-Hispanic white cases versus controls. But despite these interesting nuances, in general, the pathway and gene set enrichment analyses showed that differences in immune response-related pathways were enriched across all of the different analyses that they did, whether joint or stratified, despite the underlying gene sets. Overall, the data suggests converging pathophysiological processes in AD despite differences in ancestry. So next up is a paper focused on microRNAs, titled miRNA Profiling in Plasma Neural-Derived Small Extracellular Vesicles from Patients with Alzheimer's Disease. This one's by first author Serpent and last author Galimberti, published in Cells. So small extracellular vesicles can travel into peripheral blood from the central nervous system while carrying unique molecular markers, like proteins on their surface, that indicate where they came from, and this can be the brain. In this study, the authors set out to determine whether total plasma, or neural-derived small extracellular vesicles, and their microRNA cargo differed between patients with AD and healthy controls. So from 40 patients with AD and 40 healthy controls, they isolated the total plasma extracellular vesicles and then further fractionated the neural-derived small extracellular vesicles out based on their protein content. Then they used high-throughput advanced TaqMan miRNA open arrays to measure levels of 754 miRNAs simultaneously. They found that 3 microRNAs were significantly upregulated in the neural-derived extracellular vesicles from patients with AD, and 1 was significantly downregulated compared to controls. As far as I know, this is the first study of its kind to identify a seemingly AD-specific miRNA signature in these small extracellular vesicles. It'll be interesting to see whether this can be replicated in larger sample sizes, since it may shed some light onto disease mechanisms as well. So next up is another paper on RNA levels, but through a bit of a more computational lens, and it's titled Predicting Clinical Dementia Rating Using Blood RNA Levels. This is by first author Miller and last author Cowie, published in Genes. So as succinctly stated in the title of the paper, the group set out to use blood RNA levels to predict who is classified as cognitively normal, having mild cognitive impairment, or having probable Alzheimer's disease according to the Clinical Dementia Rating, which is a commonly used tool to measure cognitive performance. They used data from 741 participants from the Alzheimer's Disease Neuroimaging Initiative, or ADNI, database, and initially found that only levels of RNA coding for the chloride intracellular channel 1 were significantly different between the 3 groups after correction, but that these levels alone were not enough to predict cognitive status very well. Then they used machine learning to generate an improved model based on all blood RNA levels. So in combining other types of RNA that weren't individually significant between groups, they generated a model that had a receiver operating characteristic, area under the curve, also known as the ROC AUC, or the diagnostic ability of the model, of 0.904. So the best possible ROC AUC, or diagnostic ability, is 1. So 0.904 is pretty good at distinguishing between groups. So while I don't personally know much about computer science and algorithms, I think this type of study is really interesting and illustrates how machine learning will be an extremely useful tool in diagnosing and predicting Alzheimer's disease moving forward. Next up is the last paper on RNA blood biomarkers, and it's by first author Sell and last author Helmich, called Principal Component Analysis of Blood MicroRNA Datasets Facilitates Diagnosis of Diverse Diseases, and this was published in PLOS 1. This study had more of a broad scope than just Alzheimer's disease, and used a statistical method called principal component analyses to determine whether blood microRNAs have potential as disease identifiers. The group analyzed publicly available blood microRNA datasets from the Gene Expression Omnibus from the National Institutes of Health in the US by using bioinformatics software Clucor Omics Explorer. Principal component analysis was used to deal with the high dimensions of data, so the tons of different microRNAs measured, and using this the authors found the top 3 discriminating microRNAs that identified patients with specific diseases. This included Alzheimer's disease and many other diseases from healthy controls. I had a look at the full paper, and interestingly, the 3 distinguishing miRNAs weren't the same as the previous paper I talked about that isolated miRNA from the small extracellular vesicles. But the authors plan to carry on this work to generate a clinical reference database by assembling principal component and heat map analyses of existing and future blood miRNA datasets. So surely a useful tool, not only in Alzheimer's disease, but in the identification of biomarkers for other diseases as well. Okay, so moving on into the next section, I have 5 papers focused on the blood biomarker neurofilament light. So this is a pretty well-established biomarker now, and it's actually one that my lab at UBC measures pretty often in projects spanning AD, traumatic brain injury, and even COVID-19 recently. So it's ultimately a marker of neuronal, and more specifically axonal damage, since it's part of the neuronal cytoskeleton, so it really shouldn't be found in the blood unless the neurons are dying, which is a hallmark feature of AD and other neurodegenerative diseases. So the first paper up in this subsection developed a new assay to measure it in plasma, and it's called Development of an Assay of Plasma Neurofilament Light Chain Utilizing Immunomagnetic Reduction Technology. This is by first author Liu and last author Yang, published in PLOS 1. So this study developed a new kit for measuring plasma neurofilament light, or NFL, not to be confused with the Football League, using a technique called immunomagnetic reduction, where antibodies are conjugated to magnetic nanoparticles. This paper characterizes the preclinical properties of the assay, including that it had a lower limit detection of 0.18 femtograms per mil, so it's very sensitive, and a dynamic range of 1,000 picograms per mil. The authors also assessed the utility of the assay and evaluated whether it could distinguish between cognitively healthy controls, patients with Parkinson's disease dementia, and AD. Plasma NFL levels were significantly higher in all disease groups compared to the control group, with the highest being in the AD group. For distinguishing between patients with dementia and cognitively normal controls, the ROC AUC was 0.868, and for further distinguishing between AD and PD, the ROC AUC was 0.630. So when you think about it, it isn't super surprising that the AUCs were pretty low or pretty far from 1, considering that neurons are damaged in both AD and PD, so you'd expect that levels would be sort of similar, at least between the two types of dementia. But overall, this assay may be very useful in dementia screening and in the larger world of plasma biomarkers, considering the wide range of applicability of plasma NFL levels. The next paper assessed whether levels of NFL could predict features of AD, and it's titled Elevated Plasma Neurofilament Light in Aging Reflects Brain White Matter Alterations but Does Not Predict Cognitive Decline or Alzheimer's Disease. It's by first author Nyberg and last author Adolfson, published in Alzheimer's and Dementia, Diagnosis, Assessment, and Disease Monitoring. So in this paper, the authors leveraged the longitudinal Betula study that began in 1988 and sought to characterize NFL accumulation in normal aging, preclinical, and clinical AD. They measured NFL using the Quanterix Simoa NF Light Kit in 37 participants with AD, 105 participants with preclinical AD, and 126 age and sex match controls in a cross-sectional case control study. They also measured longitudinal changes in the plasma biomarker in 100 participants who had longitudinal neuroimaging data as well. In their cross-sectional portion of the study, they found significantly higher levels in AD patients compared to controls, but no significance in the preclinical group. The longitudinal portion of their study showed that there were no significant correlations between the marker and cognition, but there was with brain white matter. They also observed age-related increases in NFL levels, which is to be expected. So overall, increased neurofilament light seems to distinguish clinical AD and likely serves as a biomarker for changes in white matter. So with converging evidence, the next paper is called Serum Neurofilament Light Chain Levels Are Associated with White Matter Integrity in Autosomal Dominant Alzheimer's Disease. This is by first author Schultz and last author Gordon, published in Neurobiology of Disease. So since white matter damage is a common feature of AD, the authors investigated whether serum NFL levels were able to reflect it. They leveraged the Dominantly Inherited Alzheimer's Network Study, also known as DIAN, and they analyzed NFL levels in 117 patients with autosomal dominant mutations that lead to early onset AD, and 84 non-carriers, so controls. Importantly, a subset of 41 of the mutation carriers had longitudinal NFL and MRI data. They found that in mutation carriers, so those who would eventually develop AD, that NFL was positively correlated with white matter hyperintensity lesion volume, and that a greater increase in NFL levels was associated with many different indications of reduced white matter integrity. Importantly, they found no relationships with NFL in the non-carriers. So overall, this study provides powerful evidence of the relationship between NFL and white matter integrity in the brain. Next up is a paper called Plasma Neurofilament Light, a marker of neurodegeneration in mild behavioral impairment. And it's by first author Node and last author Ismail, published in the Journal of Alzheimer's Disease. Mild behavioral impairment is characterized by the onset of neuropsychiatric symptoms in older adults, and it's classically known as an at-risk state for cognitive decline and dementia, but its direct relationship with neurodegeneration isn't really totally understood yet. So this study set out to determine whether the condition is associated with neurodegeneration reflected by plasma NFL levels. So changes in neurofilament light were tracked over 2 years in a cohort of 584 non-demented participants from the Alzheimer's Disease Neuroimaging Initiative Study, or ADNI. They found an interaction between the time and mild behavioral impairment status that significantly predicted changes in NFL levels after controlling for age, MCI, and demographics. Overall, the results of this study suggest that mild behavioral impairment may be a clinical proxy of early neurodegeneration, as reflected by NFL levels. And it also reinforces the relevance of NFL as a biomarker reflective of cognitive and behavioral status. So last in this subsection on neurofilament light is a paper by first author Loeffler and last author Hutter-Paye, and it explores the wide range of applicability of NFL as a blood biomarker in animal models. And it's titled Neurofilament Light Chain as a Biomarker of Neurodegenerative and Rare Diseases with High Translational Value. It was published in Frontiers in Neuroscience. So it's pretty well established that plasma NFL levels are increased in any neurodegenerative disease, but whether levels reflect disease state in preclinical models isn't fully understood. So in this paper, the authors examined neurofilament light chain plasma levels and cerebral spinal fluid levels in the 5x FAD mouse model of AD and in mouse models of ALS, Parkinson's disease, and Gaucher disease. They found that NFL plasma levels progressively increased in the AD and ALS models, but didn't really change in the Parkinson's disease mouse model. So overall, it seems that the 5x FAD mouse model showed a similar trajectory to human AD changes in NFL, which may mean that it has translational value as a model of Alzheimer's disease. So now on to the next subsection, which is a group of papers that measured different lipids and lipoproteins in the plasma as reflections of AD. And first up is a paper called Association of Dyslipidemia with Alzheimer's Disease in a Cohort of Postmenopausal Women. It's by first author Lee and last author Han, published in the Journal of International Medical Research. So this study set out to determine whether there is an association between dyslipidemia and AD in a cohort of postmenopausal women, as was said in the title. So it was a retrospective study that analyzed data from 200 postmenopausal women with early AD and 180 age and sex match controls. Measures included in the study were body mass index and lipid-related parameters, including high and low-density lipoprotein cholesterols, total cholesterol, triglycerides, and a series of apolipoproteins. Using multivariate binary logistic regression, the authors found that reduced levels of HDL cholesterol and increased levels of LDL cholesterol were the only measures associated with the risk for developing AD. The next paper looked at blood biomarkers for cerebral amyloid angiopathy, or CAA, which is the deposition of amyloid beta into cerebral vessels, using a mouse model of sporadic CAA. And it's titled Platelet and Plasma Phosphatidylcholines as Biomarkers to Diagnose Cerebral Amyloid Angiopathy. It's by first author Feudal and last author Humpel, published in Frontiers in Neurology. So this study analyzed lipids including phosphatidylcholines and lysophosphatidylcholines in platelets and plasma of a novel mouse model of cerebral amyloid angiopathy to shed light on ultimately how to characterize CAA. Various lipids were significantly altered in plasma and platelet samples from cerebral amyloid angiopathy mice compared to controls. For specific results on which lipids were altered, you can refer to the paper, as I think listening to me read out a list of lipids might be kind of painful. But overall, cerebral amyloid angiopathy appears to have a unique pattern of lipids at the plasma and platelet level, and this should be investigated in humans as well. So I'll definitely be looking into the mouse model they used in this paper since my work involves CAA. But next we're back to humans with a paper titled Relationship Between Lipoprotein A and Cognitive Function, Results from the Berlin Aging Study 2, and it's by first author Rowe and last author Duzel, published in Scientific Reports. So atherosclerotic effects associated with altered lipid patterns have been implicated as a driving force in age-related cognitive decline. This study investigated the relationship between lipoprotein A and healthy cognitive aging using data from 1,380 Berlin Aging Study 2 participants. They were aged 60 years and older. The authors used regression models accounting for ApoE genotype, lipid parameters, and other risk factors to determine whether there was an association between lipoprotein A and specific cognitive functions. A gender effect was found where men in the lowest lipoprotein A quintile had better cognitive performance in tasks focused on executive functions and processing speed, but no significant results were observed in women. Overall, the results indicate that low lipoprotein A levels are associated with enhanced cognitive performance, but this association was relatively weak and further investigation is definitely required. The next paper looked at levels of apolipoprotein J, also known as clusterin, in the context of AD, and it was by first author Ha and last author Kim, titled Plasma Clusterin as a Potential Link Between Diabetes and Alzheimer's Disease. And it was published in Journal of Clinical Endocrinology and Metabolism. Plasma levels of clusterin have been associated with both AD and diabetes mellitus in the past. And this study investigated both the independent and interactive relationship of plasma clusterin in both diseases using a cross-sectional observational study of 51 participants. Participants were grouped by cognitive status measured by neuropsychological measures and MRI, and metabolic impairments measured by blood tests. Participants with AD and/or metabolic impairment had higher levels of plasma clusterin. While no synergistic effect was found, AD and diabetes mellitus had an additive effect on clusterin levels, so with both diseases, the levels were the highest. Reinforcing this triad relationship, the authors also found that the association between diabetes mellitus and AD was significant when clusterin was considered as the mediator of their relationship. Levels were also correlated with indicators of neurodegeneration and microvascular insufficiency. Overall, these analyses suggest that clusterin serves as a potential mediator and biomarker of AD and diabetes mellitus, and more work should be done with larger sample sizes to investigate this interesting relationship. Next, we have a couple of papers focused on hormones, with the first one being serum hepcidin levels in cognitively normal adults with high neocortical amyloid beta load. This is by first author Chatterjee and last author Martins, published in Journal of Alzheimer's Disease. So iron dysregulation has been found in AD along with increased serum levels of hepcidin. Hepcidin is a hormone that suppresses iron release, leading to an intracellular accumulation of iron. So this study aimed to determine whether elevated serum hepcidin levels occur in the early stages of AD, using samples from 100 cognitively normal adults, 35 of which had high neocortical amyloid beta load. They found that serum hepcidin was significantly higher in adults with high amyloid beta load, even when adjusting for age, gender, and ApoE4 carrier status. Adding serum hepcidin levels along with plasma A-beta 40:42 ratio improved the accuracy of distinguishing those with high from those with low neocortical amyloid beta loads. Overall, these results implicate increased serum hepcidin levels as a risk and potential biomarker for AD, as well as provide further support for iron dysregulation in AD. So the last paper in our lipid subsection is called Development of Classification Models for the Prediction of Alzheimer's Disease Utilizing Circulating Sex Hormone Ratios. This is by first author Hayashi and last author Atwood, published in the Journal of Alzheimer's Disease as well. Sex hormones are known to be essential for normal cognitive health. People with dysregulated hormone levels around the time of menopause and andropause are at a higher risk for developing cognitive loss and AD. So the objective of this study was to determine whether plasma and serum hormone levels could be a surrogate biomarker for cognitive decline. The authors measured 11 steroids and follicle-stimulating hormone in over 100 samples of each matrix, so both plasma and serum, from AD and control subjects from the Wisconsin Alzheimer's Disease Research Center. Due to the large variability, hormone concentrations were unable to predict AD status alone, but using classification tree analyses revealed interactions between individual hormones and hormone ratios with AD. The strongest associations included cortisol, cortisone, androstenedione, progesterone, and 17-beta-estradiol, which accurately predicted 81% of AD and 64% of control cases. These data suggest that classification tree analyses of hormone ratios may be useful in predicting potential biomarkers for AD. So moving on now to a section devoted to other proteins, starting with a broad strokes paper involving a lot of machine learning. We have a paper titled Identifying Protein Biomarkers in Blood for Alzheimer's Disease. This is by first author Zhao and last author Wang, published in Frontiers in Cell and Developmental Biology. These authors took a computational approach to identify blood biomarkers for AD and other related diseases. Their overarching hypothesis was that similar diseases share similar underlying proteins. They developed an iterative computational method based on disease similarity, and 39 diseases that are similar to AD were chosen to be included in this analysis. Overall, they had a pool of over 1,000 different proteins based on these diseases. So their method, which I don't fully understand, again because I'm not deep into computer science, identified 284 proteins that may be related to AD, which can be compared to the 184 from the pool that they started with that are already reported in databases like UniProt. They cross-validated their list of 284 using case studies and found an AUC of 0.9251, so that's the diagnostic ability of their model, including all these proteins. And importantly, of their 100 novel proteins identified, 7 are specific to blood, including a couple that I recognized, like prothrombin and fibronectin. Overall, the study presented a new method for prioritizing AD-related proteins, which may be useful biomarkers. Another lovely illustration of the value of machine learning for mining the incredible amount of data that is already out there. Next up, we have a paper called TDP43 is elevated in plasma neuronal-derived exosomes of patients with Alzheimer's disease. This is by first author Zhang and last author Gordon, published in Frontiers in Aging Neuroscience. The DNA binding protein TDP43 has been identified as a common proteinopathy in aging and a neuropathological comorbidity in AD patients. However, further in vivo examination of TDP43 in AD has been difficult due to its low concentration in cerebrospinal fluid. This study used an ELISA-based method to determine TDP43 levels in plasma neural-derived exosomes in 24 amyloid PET-confirmed AD patients and 15 healthy controls. TDP43 was increased in AD patients, however no associations with cognition, neuropsychiatric symptoms, or ApoE genotype were found. These results indicate that TDP43 accumulation can be measured in AD patients from plasma neural-derived exosomes, which is promising for future investigation of TDP43's role in AD. Next up, we have a paper that used a technique that I hadn't heard of, and it's called Indicators of Rapid Cognitive Decline in Amnesic Mild Cognitive Impairment, the Role of Plasma Biomarkers Using Magnetically Labeled Immunoassays. This is by first author Tsai and last author Yang, published in the Journal of Psychiatric Research. This study used a sensitive technique called immunomagnetic reduction, which leverages magnetic nanoparticles conjugated to antibodies to measure analytes in solution. In particular, this group aimed to figure out whether plasma levels of common protein aggregates such as amyloid, tau, and alpha-synuclein were associated with rapid cognitive decline in patients with amnesic cognitive impairment and AD. Their study measured the analytes with immunomagnetic reduction in 19 patients with AD, 24 patients with amnesic MCI, and 20 healthy controls. Each participant was also assessed with the MMSE twice, with about 1 year in between, to track cognitive decline. The group found that higher plasma levels of A-beta-42 and alpha-synuclein were able to identify cognitive decline in patients with amnesic MCI with an area under the curve of 0.7, indicating that these may be useful plasma biomarkers to track cognitive decline. However, due to the very small sample size, these results should be validated in the larger cohorts. The next paper evaluates a series of matrix metalloproteinases, which are enzymes that can degrade extracellular matrix components like collagen, and to my understanding may play roles in degrading amyloid and a series of other proteins as well, making them a hot topic in diseases involving protein aggregation. So this paper is titled Influence of Plasma Matrix Metalloproteinase Levels on Longitudinal Changes in Alzheimer's Disease Biomarkers and Cognitive Function in Patients with Mild Cognitive Impairment Due to AD, registered in the Alzheimer's Disease Neuroimaging Initiative database. Nice long title for us. So this is by first author Abe and last author Hishimoto, published in Journal of Neurological Sciences. The authors of this paper set out to determine whether levels of matrix metalloproteinases, or MMPs, in plasma were associated with longitudinal changes in AD-related features, including CSF biomarkers, brain atrophy, and cognitive function. To do this, they used a group of 95 ApoE4 carrying patients from the ADNI cohort who had mild cognitive impairment due to AD, indicated by their confirmed AD-specific CSF biomarkers, as well as a group of controls. They obtained baseline data of 6 different MMP or MMP-related proteins in plasma and longitudinal data on a series of AD-related features over 4 years, and used this data to build linear mixed regression models. They found no significant differences between levels of baseline plasma MMPs in their MCI-AD group compared to their control group, and no significant correlations between MMP levels and longitudinal changes in CSF biomarkers. However, interestingly, they did find that declines in hippocampal volumes and cognitive function were significantly faster in MCI-AD patients who had high MMP9 levels at baseline. This is in line with a lot of evidence pointing to the involvement of MMP9 in AD, and I'm really interested to see whether these findings can be replicated in another cohort. Next up, we have a paper called Proteomic Profiles for Alzheimer's Disease and Mild Cognitive Impairment Among Adults with Down Syndrome Spanning Serum and Plasma, an Alzheimer's Biomarker Consortium Down Syndrome, or ABCDS study. This is by first author Peterson and last author O'Brien, published in Alzheimer's and Dementia, Diagnosis, Assessment, and Disease Monitoring. So as a bit of a preface to this one, given that Down syndrome is caused by trisomy 21 and the amyloid precursor protein is on chromosome 21, people with Down syndrome often develop considerable A-beta plaque loads as they age, and many also develop the cognitive dysfunction characteristic of Alzheimer's disease as well. So this makes people with Down syndrome uniquely poised to provide insight into AD. So building on this study used previously generated serum and plasma proteomic profiles from adults with Down syndrome to determine whether they could accurately discriminate between those with MCI, AD, and cognitively stable controls. The group analyzed data from 305 participants enrolled in the Alzheimer's Biomarker Consortium Down Syndrome, or ABCDS, study, and impressively they found high sensitivity and specificity in their models, and that the proteomic profile was able to distinguish between MCI and cognitively stable participants with an AUC of 0.95. Similarly, it was able to distinguish between AD and cognitively stable controls with an AUC of 0.93. Some of the main proteins driving the discriminative capacity were eotaxin 3, interleukin 10, C-reactive protein, IL-18, and serum amyloid A. And these proteins were detected at nearly the same levels in either serum or plasma, so it didn't matter which matrix they were measured in. The next paper is by first author Scott and last author De Riviero Vaccari, and it is called The Inflammasome Adapter Protein, ASC, in Mild Cognitive Impairment and Alzheimer's Disease. And it was published in the International Journal of Molecular Sciences. This group aimed to identify novel early markers of MCI with a focus on levels of the two inflammasome signaling proteins, ASC, also known by the long painful name of apoptosis-associated speck-like protein containing a caspase recruitment domain, and IL-18. They measured the levels of these two analytes as well as soluble APP, alpha and beta, and our old friend NFL. all in the serum of patients with MCI, AD, and healthy age-matched controls. They found that levels of ASC were higher in patients with MCI than in patients with AD, and that levels in both cognitively impaired groups were higher than in controls. The AUC for discrimination between MCI and healthy controls was very high at 0.974, which was comparable to the discriminative capacity of soluble APP and higher than NFL, which only had an AUC of 0.77 here. They found similar results in AD versus controls and MCI versus AD, indicating that ASC may be a promising biomarker. The abstract didn't mention anything about their IL-18 results, but I'm assuming they weren't as exciting if they didn't make the cut. But check out the paper for more info. The next paper is titled Serum FOXO3A, A Ray of Hope for Early Diagnosis of Alzheimer's Disease. This is by first author Pradhan and last author Day, published in Mechanisms of Aging and Development. So FOXO3A is a transcription factor expressed in many different cell types, but that is found in cells in the inferior temporal and parahippocampal regions of the brain. Given this localization to regions closely tied to cognition, the group set out to determine whether it could be a useful plasma biomarker for AD. The group measured serum FOXO3A through surface plasmon resonance with an antibody specific for the protein in patients with AD, MCI, and in geriatric controls. They found that serum FOXO3A was significantly different between all three groups, with the lowest levels being in AD. The AUC for AD versus MCI was 0.84, MCI versus control was 0.98, and AD versus control was a nice crisp 1.0, which outperformed phospho-tau 181 and total tau. While these are impressive values, it should be noted that the study only enrolled a total of 49 subjects, and these results need to be validated in another larger cohort. Next up, we have a mouthful of a title. It's called The CNS-Specific Proteoglycan Brevican, and its ADAM-TS4 cleaved fragment showed differential serological levels in Alzheimer's disease, other types of dementia, and non-dementia controls, a cross-sectional study. This is by first author Janesco, last author Henriksen, published in PLOS 1. Brevican is a brain-specific proteoglycan found on neurons and has been shown to be proteolytically cleaved during neurodegeneration. In this study, the authors developed ELISA-based assays to measure Brevican fragments, including the N-terminal, or N-brev, and the ADAM-TS4 generated fragment, or brev-A, from serum and determine their potential to discriminate those with dementias from controls. To evaluate their assays, they applied them to serum from 28 patients with AD, 48 patients with other types of dementia, and 48 patients with non-dementia-related memory complaints. Patients with non-AD dementias had lower levels of NBREV and higher levels of brev-A compared to controls and AD patients. Somewhat unexpectedly, there was no difference in the levels of either fragment between AD and controls. When the data from the two fragments were combined as a ratio of NBREV over brev-A, similarly to the amyloid beta fragment ratios commonly used, the differences observed between the non-AD dementia group and other groups was more pronounced, yielding AUCs of 0.75 for discrimination between non-AD dementia and controls, and 0.72 for discrimination between non-AD dementia and AD. This suggests that these assays may be useful in distinguishing between types of dementia, although more work needs to be done to parse apart their non-AD dementia group. That caps off the blood biomarkers targeting proteins, and now we're on to the final catch-all section of everything else. So starting off with serum uric acid, Alzheimer's-related brain changes, and cognitive impairment. This is a paper by first author Kim, last author Lee, published in Frontiers in Aging Neuroscience. Reduced serum uric acid is associated with AD and related cognitive impairment, but how it actually contributes to specific pathological mechanisms is unknown. In this study, the authors set out to determine whether relationships exist between serum uric acid and features of AD, such as cognitive deficit, cerebral A-beta and tau deposition, glucose metabolism, and white matter hyperintensities. Their study enrolled 430 non-demented older adults, in which they measured serum uric acid levels and a series of features of AD. From all of these features they measured, they found that serum uric acid was only significantly associated with cerebral glucose metabolism, and that other cognitive-related associations disappeared when cerebral glucose metabolism was controlled as a covariate, indicating that the glucose metabolism mediated the effect. Overall, these findings suggest that there's an association of low serum uric acid with AD-related cerebral hypometabolism, but the causality of this relationship is still unclear. Next is another mouthful of a title. It's called Mini Nutritional Assessment May Identify a Dual Pattern of Perturbed Plasma Amino Acids in Patients with Alzheimer's Disease, a Window to Metabolic and Physical Rehabilitation. This is published in the journal Nutrients by first author Aquiliani and last author Veri. So far, there are conflicting results about alterations in plasma amino acid levels in patients with AD. This study aimed to clear the muddy waters a bit and provide a more homogeneous amino acid profile for patients and examine the correlations between these profiles and cognitive status. The group measured venous plasma amino acids using an HPLC-based method in 54 patients with AD and 17 matched cognitively healthy controls, who were all stratified for nutritional state using the Mini Nutritional Assessment. They found that patients with AD had significantly lower levels of aspartic acid and taurine and higher 3-methylhistidine, all of which were independent of their nutritional state class. They also found that serine and histidine levels were positively correlated with verbal memory and attention capacity deficits, respectively, and that total amino acids negatively correlated with verbal attention capacity deficits. Next up is a shift to epigenetics with the paper Harnessing Peripheral DNA Methylation Differences in the Alzheimer's Disease Neuroimaging Initiative, or ADNI, to reveal novel biomarkers of disease. This is by first author Vasan Thakumar and last author Saikin, published in Clinical Epigenetics. This study evaluated differentially methylated positions, or DMPs, as novel blood-based biomarkers for AD in peripheral blood samples. For their study, this group leveraged the ADNI consortium and chose a subset of the cohort with a comprehensive dataset including imaging, genetics, and transcriptomics. They used Illumina technology to identify differentially methylated positions in the peripheral blood samples from a total of 653 individuals who had either AD, MCI, or were age-matched controls. They found that the most highly significant differentially methylated loci between groups correlated with cognitive function. Interestingly, they also found that DMPs were enriched in genes such as BDNF, BIN1, and ApoC1, all of which are known to be related to neurodegeneration. This paper contains a ton of data, and if you have the time or this is your field, you should definitely comb through it. It's also a very impressive and valuable addition to the ADNI database. Overall, this study demonstrated that peripheral differential methylation exists between patients with AD and age-matched controls, which provides opportunities to delve into whether differential peripheral methylation can be a biomarker for AD. Next in this mixed bag is a paper titled Impact of Serum Calcium Levels on Alzheimer's Disease, a Mendelian Randomization Study. It's by first author He and last author Liu, published in the Journal of Alzheimer's Disease. Changes in calcium homeostasis are thought to be an underlying mechanism in AD, but whether serum calcium levels are genetically associated with AD is not yet clear. In this study, the authors aim to establish a causal link between serum calcium levels and AD using Mendelian randomization. They leveraged a large-scale serum calcium genome-wide association study, or GWAS, dataset of over 60,000 individuals, and a large-scale AD GWAS study of nearly 20,000 patients with AD and nearly 40,000 controls. Using the inverse variance weighted as the main analysis method, they found that increased calcium levels were significantly associated with decreased risk for AD, with an odds ratio of 0.57 and a 95% confidence interval of 0.35 to 0.95. These data provide support for further investigation into the causal nature of increasing calcium intake to reduce the risk of developing AD. And finally, our last paper in this episode is called Extracellular Vesicle Biomarkers of Alzheimer's Disease Associated with Subclinical Cognitive Decline in Late Middle Age. It was published in Alzheimer's and Dementia by first author Aaron and last author Kapogiannis. So we've already heard about the miRNA profiles of these vesicles in a previous paper in this episode, but this group focused on measuring tau and insulin signaling biomarkers in the vesicles isolated from serum, to determine whether they could predict cognitive decline. The case control study used repeated serum samples from 73 cognitively declining and 73 cognitively stable patients from the Wisconsin Registry for Alzheimer's Prevention, and isolated the neuronal extracellular vesicles through immunocapture. They found that individuals who were cognitively declining had higher baseline total tau, p-tau213 and p-tau181, and higher annualized changes for the insulin signaling markers PIR and PIGF-1R, indicating that insulin biomarkers may prospectively predict cognitive performance. Combining the biomarkers in a model yielded an AUC of 0.94 in a training model for distinguishing between those who cognitively declined and those who were stable, and an AUC of 0.75 when it was applied to an independent test dataset. Ultimately, in this paper, these data hold promise for future evaluation of biomarkers from extracellular vesicles. And that's all for this episode. I hope you learned as much as I did curating it for you, and I'll be back soon with more episodes. As always, thanks for listening.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and a member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.